Hello guys, so welcome to EasyMed, and so today's our topic is osmosis and tonicity. So osmosis, what is osmosis? Osmosis is defined as the movement of the water from lower concentration to higher concentration through semi-permeable membrane. So when two solutions were separated by a semi-permeable membrane, then there is movement of water from low concentration to higher concentration, and this process is called osmosis. And there are two terms, osmolarity and osmolality. Osmolarity is the osmoles per liter of water, and osmolality is osmol expressed in per kg of water. So what is osmol? So osmol is defined as the 1 gram of molecular weight of any solute divided by number of ions or number of dissociated ions. Let's weight equal to 180 gram. So number of dissociated ion equal to single. So when glucose is placed in water, there is no dissociation. So 1 osmol of glucose equal to 1 gram molecular weight, that is 180 gram, divided by number of dissociated solute, that is 1 equal to 180 gram. So 1 osmol equal to 1 molecular weight. In case of NaCl, NaCl is dissociated into 2 ions. So 1 osmol of NaCl equal to 1 gram in putting formula 1 gram molecular weight of solute divided by number of dissociated ions, so 58.5 gram divided by 2. And now let's talk about tonicity. So tonicity is a comparison of a two solution in term of osmolarity. So on the basis on comparing two solutions, they may be isotonic or hypertonic, or they may be hypotonic solution. So if this is basically comparison. So two solutions were needed and we compare them. And in case of isotonic solution, when two solutions were separated by semi-permeable membrane, so there is movement of water. But in case of isotonic solution, there is equal osmolarity, that is osmolarity of outer solution equal to osmolarity of inner solution. So there is equal movement of water or no net movement of water. If 50 ml goes in, then 50 ml comes out, so no net movement. So there is no size change of the both cells. So in case of hypertonic solution, when one solution is having more osmolarity than the another solution, then the higher solution is called hypertonic solution. So in case of in the picture, there is the outer solution has the higher osmolarity, so this is hypertonic solution. And so there is, when there is difference in the osmolarity of the two solutions, so there is net movement of the water. So in this case, there is the movement of water from lower concentration to higher concentration, that is from inner cell to outer cell. So the inner cell size decreases and outer cell size increases because the water moves from inner cell to outer cell. So in case of hypotonic solution, so when one solution have the lower osmolarity than another cell, then the lower osmolarity cell is called hypotonic solution. Okay, so in this picture, the outer solution have lower osmolarity, so this is hypotonic solution. So when there is a difference in osmolarity between two solutions, so there is movement of water. So movement of water is always from lower concentration to higher concentration. So there is movement of water from outer cell to inner cell. So there is movement from the outer cell to inner cell. So the outer cell size decreases and inner cell size increases as water moves towards inner cell. So let's discuss our questions. Our first question, a cell is placed in a hypertonic solution, the size of cell will be, okay, there is one cell which is placed in a hypertonic solution. So there is net movement of water. So water is moved from inner cell to outer solution, so there is decrease in cell size. And the second cell, the human cell is placed in 0.9% NaCl solution. Result of the experiment will be, okay, so 0.9% NaCl is actually the isotonic with the human cell. So there is isotonic, both are isotonic solutions, so there is no net movement, so the cell size remains constant. Okay, 1 gram molecular weight of glucose contains, okay, 1 osmol equal to molecular 1 gram molecular weight divided by dissociated ions. So when glucose is placed in water, there is no dissociation, that is one single ion is present. So 1 osmol equal to 1 molecular weight divided by dissociated ion, that is the single ion. So this is 1 osmol equal to 1 molecular weight. In case of NaCl, it is dissociated into 2 ions, that is Na plus and Cl minus. So 1 osmol equal to 1 gram molecular weight divided by number of dissociated ions, that is 2. So 1 osmol equal to 1 gram molecular weight divided by now 2 ions, so Na plus and Cl minus. So 1 gram molecular weight equal to 2 osmoles. So thank you for watching.